Hi everyone, I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and subscribe the channel. In the last session, we ended up with energy yield of glycolysis. In today's session, we will start with regulation of glycolysis. Here we will see regulatory enzymes of glycolysis, in which there are activators and inhibitors of the enzyme. Then factors regulating glycolysis, in which we will see the comparison between hexokinase and glucokinase. Then we will see in RBCs how fructose 2,6 bisphosphatase differs from other cells. Then we will see cori cycle, in which lactate is produced from glucose inverses and then transferred to the liver. Then lastly we will see pyruvate dehydrogenase complex. Good morning everyone. Let us start the regulation of glycolysis. The regulatory enzymes for key enzymes for glycolysis are glucokinase or hexokinase, that is step 1, phosphofructokinase, that is step 3, and pyruvate kinase, that is step 9. Here are certain regulatory enzymes of glycolysis which are activated and inhibited. Hexokinase is inhibited by glucose 6-phosphate. Glucokinase is activated by insulin, whereas it is inhibited by glucagon. Phosphofructokinase, it is activated by insulin, AMP, fructose 6-phosphate, phosphofructokinase 2, and fructose 2,6 bisphosphate, whereas it is inhibited by glucagon, ATP, citrate, low pH, and cyclic AMP. Then we have pyruvate kinase, which is activated by insulin and fructose 1,6 bisphosphate, whereas it is inhibited by glucagon, ATP, and cyclic AMP. PDH or pyruvate dehydrogenase complex. It is activated by coenzyme A and NAD, whereas it is inhibited by acetyl CoA or NADH. Glucokinase or hexokinase. Phosphorylation of glucose by these enzymes is a reaction which is regulated by feedback inhibition. Example, hexokinase is inhibited by glucose 6-phosphate, whereas it is activated by insulin. For example, glucokinase is induced by insulin. Glucokinase is active mainly in liver, has a high Km for glucose and low affinity. Hence, glucokinase can act only when there is adequate glucose supply, so that excess can be stored. Hexokinase with low Km and high affinity can phosphorylate glucose even at lower concentrations, so that glucose is made available to brain, heart, and cardiac muscles. Glucokinase can act only when there is plenty of glucose. Thus, when the supply of glucose is limited, glucose is made available to brain and muscles. Second enzyme is phosphofructokinase, that is PFK. It is the most important rate limiting enzyme for glycolysis pathway. In this diagram, we can see ATP generated at different stages. First, it is involved in generated in 1,3 bisphosphoglycerate to 3,3-phosphoglycerate, and the second one is phosphoenol pyruvate to pyruvate. ATP and citrate are the most important allosteric inhibitors, whereas AMP acts as an allosteric activator. The next enzyme is PFK2 or phosphofructokinase 2. Fructose 2,6 bisphosphate increases the activity of phosphofructokinase. Fructose 2,6 bisphosphate is formed from fructose 6-phosphate by the action of an enzyme called PFK2 or phosphofructokinase 2, which is not equal to phosphofructokinase 1. Fructose 2,6 bisphosphate is hydrolyzed to fructose 6 phosphate by enzyme fructose 2,6 bisphosphatase. The activities of both the enzymes PFK2 and fructose 2,6 bisphosphate phosphatase are reciprocally regulated. When glucose supply is in plenty, PFK2 is dephosphorated and activated. So fructose 2,6 bisphosphate concentration increases. This in turn activates PFK. This thus glycolysis is favored. In this diagram, you can see fructose PFK2 or phosphofructokinase 2 and fructose 1,6 bisphosphate are reciprocally regulated. First, fructose 6 phosphate is converted into fructose 2,6 bisphosphate with the help of enzyme PFK2. Then, this 2,6 bisphosphate is converted into fructose 1,6 bisphosphate by enzyme phosphofructokinase 1. On the other hand, this fructose 1,6 bisphosphate is reversibly regulated as fructose 6 phosphate in the presence of enzyme fructose 1,6 bisphosphatase. The next enzyme is pyruvate kinase. It catalyzes an irreversible step and is a regulatory enzyme of glucolysis. When energy is plenty in a cell, glycolysis is inhibited. Insulin increases its activity, whereas glucagon decreases its activity. Pyruvate kinase is active in phosphorylated state. In this diagram, we can see that pyruvate kinase, when phosphorylated or inactive, it inhibits glycolysis, whereas when pyruvate kinase is dephosphorylated or active, it favors glycolysis. Insulin favors glycolysis by acting above three glycolytic enzymes, whereas glucagon and glucocorticoids inhibit glycolysis and favor gluconeogenesis. Coming to the next point, Cori cycle or lactic acid cycle. Definition: it is a process in which glucose is converted to lactate in the muscle, and in the liver this lactate is reconverted into glucose. It is an active contracting muscle. In an actively contracting muscle, pyruvate is reduced to lactic acid, which may tend to accumulate in the blood muscle. In this diagram, we can see Carl Cori and Gerty Cori, who proposed the Cori cycle. In liver, first lactate is converted to glucose. This glucose enters the muscle through the blood, and then it is reversely lactate is formed, and then lactate is once again reaches the liver through blood, and this whole thing is known as Cori cycle. The muscle cramps, often associated with strenuous muscle exercise, are thought to be due to lactate accumulation. To prevent the lactate accumulation, body utilizes Cori cycle. This lactic acid from muscle diffuses into the blood. Lactate then reaches the liver, where it is oxidized to pyruvate. Then it is channeled to gluconeogenesis. Regenerated glucose can enter into blood and then to muscle. This cycle is called Cori cycle. Coming to significance of the Cori cycle. The lactate produced in the muscle is efficiently reutilized by the body, but this is an energy consuming process. During exercise, lactate production is high, which is utilized by liver to produce glucose. This process needs ATP in significant quantities, which is provided by increased metabolism. This leads to increased oxygen consumption. This is the explanation for oxygen debt after vigorous exercise. and under aerobic conditions, glycolysis is inhibited. This inhibitory effect of oxygen on glycolysis is known as Pasteur effect. In the erythrocytes, coming to Rappaport Lubering cycle or BPG shunt. In the erythrocytes, step 6 of glycolysis is bypassed. This bisphosphoglycerate mutase converts 1,3 bisphosphoglycerate to 2,3 bisphosphoglycerate. Then BPG phosphatase removes the phosphate group from 3 phosphoglycerate. In this diagram, we can see BPG shunt where 1,3 bisphosphoglycerate is converted to 2,3 bisphosphoglycerate with the help of enzyme mutase. Then this 3 bisphosphoglycerate is converted into 3 phosphoglycerate with the help of enzyme phosphatase. The 2,3-BPG combines with hemoglobin and reduces the affinity towards oxygen. So, in the presence of 2,3-BPG, oxyhemoglobin will unload oxygen more easily into the tissues. Under hypoxic conditions, the 2,3-BPH concentration in the RBCs increases, thus favoring the release of oxygen to the tissues even when PO2 is low. The compensatory increase in 2,3-BPG in high altitudes favors oxygen dissociation. Hence, BPG is increased in fetal circulation. In the shunt pathway, no ATP is generated. In this diagram, we can see RBCs versus other cells. In RBCs, this 1,3 bisphosphoglycerate is converted to 2,3 bisphosphoglycerate with the help of enzyme mutase. Then this 2,3 bisphosphoglycerate is converted into 3 phosphoglycerate with the help of enzyme phosphatase. On the other hand, 1,3 bisphosphoglycerate is converted into 3 phosphoglycerate by the enzyme biphosphoglycerate kinase. Coming to metabolic fate of pyruvate. Under aerobic conditions, pyruvate is converted to acetyl-CoA which enters the TCA cycle to be oxidized to CO2. ATP is generated. Glycolysis is taking place in cytoplasm, so therefore pyruvate is generated in also cytoplasm. This is transported into mitochondria by a transporter, pyruvate transporter. Coming to pyruvate dehydrogenase complex. Inside the mitochondria, pyruvate is oxidatively decarboxylated to acetyl-CoA by pyruvate dehydrogenase. It is a multi-enzyme complex with 5 coenzymes and 3 apoenzymes. The coenzymes needed are thymine pyrophosphate, coenzyme A, FAD, NAD+, and lipoamide. The lipoic acid, otherwise called thioctic acid, has 2 sulfur atoms and 8 carbon atoms. It can accept or donate hydrogen atoms. The enzyme part of the PDH complex is made up of 3 component enzymes. The first enzyme is pyruvate dehydrogenase. It catalyzes oxidative decarboxylation. TPP is required in this step. So, thymine, which is a B complex group vitamin, is essential for utilization of pyruvate. The 2 carbon unit remains attached to the enzyme as hydroxyethyl thymine pyrophosphate. The second enzyme required is dihydrolipoyl transacetylase. Then the hydroxyl group is oxidized to form an acetyl group and then transferred from TPP to lipoamide to form acetyl lipoamide. The third enzyme required is dihydrolipoyl dehydrogenase. The last step is the oxidation of lipoamide. At the end of the reaction, the factors namely TPP, lipoamide and FAD are regenerated. Here ends our session. Kindly like and share and subscribe the channel. Thank you.